Hello students, today we are going to start with one muscle cell. Okay, so I already told you that the muscle cells, they are also known as the muscle fiber. So don't confuse in between muscle fiber and the cell. It is itself the fiber. So here this point you have to know. What is the length and diameter of the one muscle fiber? That if it is the length, it will around 3 centimeter in length, and if it comes to the diameter, then it can vary from 10 to 100 micrometer. Now let's just see that if it comes to one muscle fiber, then it will include different organelles, cell organelles like the sarcoplasm, then the sarcoplasmic reticulum, and or also the sarcolemma, including one proteinaceous filament, and that filament is known as the myofibril. So let's just see, suppose this is one muscle fiber, okay, this is one muscle cell, right? So in that muscle cell, what we can see that alternate light and dark bands are present, okay? So I told already that whenever plain polarized light is allowed to pass through one cell, then it will be seen that alternately light and dark bands are appeared, okay? Now we have to see that inside that muscle cell, what we can see, one very important organelle that is known as the myofibril. So this myofibrils, okay, they are present like this. If you need to see only one myofibril, okay, then what we can able to see, the alternate light and dark band, right? So this is the myofibril, got it? Okay, and this one, this is the muscle cell, and muscle cell or the fiber, right? Now what we have to do, we have to zoom only that one myofibril, okay? We are not even talking of one cell, we are going to see only this part, okay? So this section we are going to see now, understand? So whenever we see this myofibril's only one part, then how actually it appears, right? So just see, whenever we see only one myofibril, then it seems like, see, there are structures like this, okay? So it will appear like this. Once again, if you see another unit, then it will appear like this once again. Okay, so this is the zoomed view. Whenever you see this structure, then yes, it appears like alternate light and dark bands, but that part, length, or that region where the dark bands are present, sometimes it can be thin, sometimes it can be thick, okay? So just see what are those bands, right? So whenever we talked about the light and the dark band, see how it is, this one, okay, this is the light band, okay? Next one, you can see from here to here, right, it is the dark band, right? Here from this to this is the light band. This will be from here to here, this is the dark band. Once again, okay, then once again it will proceed, understand? Now just see here, this region, this is one dark region actually are there. Actually in this light band region, we are going to have one type of protein, right? And what is that protein? That is known as the actin protein. Got? And in this dark band region, we are going to have the myosin protein. Then we have the actin, then myosin, then actin. This way it will proceed. Okay, from here to here we are going to have myosin. From here to here we are going to have the actins, right? So just see what is the functional unit of one muscle. Okay, muscle contraction, functional unit which is going to contract now. This will be from half of the light band to the half of the next light band. Okay, so this is the whole light band. We are going to divide into two parts, this part and this part, right? So from the half of the light band to the half of the next light band, okay, and the complete dark band, it will include, this will be the sarcomere. Understand? And this sarcomere is the functional unit of the muscle which is going to contract, right? Contract and relax. Okay, so from here to here, this is one sarcomere. From here to here, this is another sarcomere. So let us see what is that sarcomere. Sarcomere will be from this to this, right? So this is the sarcomere, right? Okay, so that sarcomere will include how many light band and how many dark band, right? So just see, this question had also come that how many I bands and how many A bands form one sarcomere, right? So what is a sarcomere? It is the functional unit of muscle that undergo contraction, right? What is the length of one sarcomere? The length of one sarcomere can vary from 2 to 3 micrometer, right? So this is the length. Okay, next one thing also we need to know that this sarcomere include from here, this is the I band or the light band, right? So we have taken half of the I band, so this is the half of the I band, right? So we can call this is the half of the I band, one complete A band, this is the complete A band, one A band. And from this part, one, that means half of the I band once again. Okay, so that means how many I bands and how many A bands form one sarcomere? That will be one I and one A band, right? So this is another very much important point, that how many I bands and how many A bands form one sarcomere. Okay, so from here to here, otherwise light band is this, dark band is this, light band is this, dark band is this, right? So from this light band we are taking half of the light band, right? This one will be half of the I band also, we can say. This is dark band, that means complete A band, and from here half of the light band. So half I, one A, half I. Okay, half, one I and one complete A forms the sarcomere. Okay, so this is one very unique important point you need to know. From here a number of questions had come in need, and it is very much important also. It will come necessary to know. Okay, now we will see that in this sarcomere how actually contraction will occur, right? So we are going to concentrate on only one sarcomere, and I will tell you actually how contraction will proceed, right? So from where we are going to start, see, from this to this, right? So we are going to start from half I band, and from here half another I band. Okay, and we will draw inside one A band. Okay, so just see, this is the I band, right? Okay, done. Now in this eye band, what we know that this eye band is divided by whom? A very small thin dark band is there or not? Dark band means it's a line actually. So we'll draw this line. So this is the line actually. So this line I'm going to draw now. Okay, so I'm completely have drawn this light band. Okay, now in this light band, what we have, this line and this line. So this line I'm going to draw. This one is this. Got? And from here also, this is the line. So that means it is going to divide the eye band into two parts, right? Half of the eye band is here and half of the eye band is on this part, right? So what is this? This is the Z line. Remember, so space between two Z line is known as one sarcomere. Okay, so it is the space between two Z line. Okay, remember, it's very much important point. Now in this region, what you are going to see? We are going to see the dark band, and in this, okay, one thing, see, in this dark band, what we are going to draw now? The dark band, right? So this is going to be the dark band. Right? So this is very much important point you need to know. Now wherever, so let's just increase this part so that I can show properly. Okay, so just see. So in this region, how actually in the dark band, in the middle we will see a light region. Okay, so just see here in this light band. Okay, so this is what? This is one light band. So from this region to this region, this is the light band. Okay, so in the light band, what we are going to have? One type of protein. So this protein filament, what we can say, this is known as the actin filament. Remember, actin filament forms the light band. And this red colored lining, what we can say, this one is the dark band, where this dark band is made up of what? This dark band is made up of another protein, and that is known as the myosin. Okay, myosin is present in the dark band and actin is present in the light band. Okay, now just see that in the light band, see in this light band, one thick lining, one line we have seen. So what is this? This is the Z line, right? In this dark band, we will see us two bands here. Okay, so what is this and what is this? And in the middle, we have seen a light region. So just see wherever, okay, wherever we have the overlapping region of actin and myosin, that appears dark, right? So that appears dark. This is actually this part. In this region where the myosin is not overlapped by the actin, so this is the myosin. From here, half of the myosin is overlapping this actin or overlapping this myosin or not. So that's why what happened, this from this to this, here only myosin is present. So wherever only myosin is present, this is this part. This region is where we are going to see both actin and myosin. So here both actin and myosin is present, this one. Okay, here only myosin is there, this one. Okay, so this blue color, this green colored spot. Okay, so this is the green colored spot where you can see only the myosin. Got it? Understand? And once again, you just see another band you will find in the dark band itself. What is that? In this region, what we are going to have? Once again, overlapped actin and myosin. Okay, so here the myosin and actin, both these two are overlapping. Okay, so this will be the structure. Okay, hope you have understood. See clearly, this is very much important. Okay, so this is one Z line, this is the another Z line, right? This is the light band, this is the light band. In the light band we see a Z line, so this Z line is this Z line, right? Here this is one another light band, so where we can see a Z line, so this is the another Z line. In the middle we will find the dark band, where the dark band is divided into two parts. In the middle we have once again a light part, right? So wherever we have overlapped actin and myosin, what we can see, it appears more dark, it appears more dark in this region, it appears more dark, right? And in this region, okay, in this part, what we can see, in this part and this part, what we can see, only myosin, right? So as only myosin is present, so it appears little light. Got? Understand? Okay, so this is the structure. Now just see whenever we see the actin is actually bundled by what? Z line, right? So is myosin is somewhere hanging? It has not to be hanging, isn't that? So what happened? This myosin have to be once again bundled, right? By whom? By one another lining. So here I am drawing this line. Okay, this line and this line. So what is this? This is going to be one another line. So this is known as the M line. Okay, remember that this is the M line. Now, wherever you see only the myosin, okay, the green line region where you can see only the myosin, this green region, from here to here, okay, from this to this, what we can see, what we can tell, this is known as one zone, this is known as H zone or Hansen's zone. Okay, you can remember only as H zone. Okay, next one, wherever we have overlapped actin and myosin. Okay, we have overlapped actin and myosin here and overlapped actin and myosin here. So overlapped actin and myosin, this region and this region, this will be known as the O zone, right? Okay, so that is all about, you need to know about one sarcomere. So sarcomere, once again I am telling you, sarcomere is actually a light band and one light band and one dark band. Light band means it's not one light band, half of one light band and half of another light band, right? Half of one light band and half of one light band, right? Now the next one, that it will include a complete A band. Okay, so this is the complete A band where a protein is there, myosin, and in the light band we have another protein that is known as the actin, right? Okay. Now you just see this myosin, somewhere it is overlapped by actin. Actin and myosin are found together overlapping each other, right? So this region, this is the region, okay, in the dark band. What we can call it, this is known as the O zone. Wherever only myosin is there, this is the green dotted line, okay, green dotted space, that will be known as the H zone. And once again we will find another O zone. Okay, now like this actin is bundled by one line called as Z line. Similarly, in case of the myosin also, there will be another line. Okay, and what is that? That will be known as the H zone. Okay, now just see, we will have the contraction. So let us see, this is the relaxed state. Okay, so this is the relaxed state. Okay, this is the relaxed state. Now, whenever it will undergo contraction, okay, whenever, I can write like this also, if it undergo contraction, then what will happen? The Z lines that will come closure. Okay, so here we have the Z line, right? So Z lines are present in at a distance. Yeah, what is that? Around 2 to 2.5 or 3 micrometer. Now it will undergo contraction. So Z lines are coming closure. Okay. So just see actually how these Z lines are coming closure, because actins are coming closure, that's why. Okay, so this actins, the actin will not going to decrease its space. Okay, so actin will just come too much closer towards the M line, right? Why this is happening? Because the Z lines, they will contract. Okay, so Z lines are contracting. So whenever the Z lines are contracting, what will happen? This which zone will disappear and which zone will maximum, you have to tell me. Okay. So you just tell me that which zone is going to be decreased. Okay. So just see here, this myosin will remain same. Okay. It's not like that myosin will decrease or something will happen. No. Okay, so this myosin length will remain same, or it may be a little decrease, but it remains same. Okay, and the Z lines are coming together from both the sides in opposite direction towards the M line. Okay, and after that it will have maximum contraction. After that it will not have contraction like this. Actins from both the sides will come closure, and this is the maximum contraction. After that there will be no contraction. Okay, no further contraction can happen after this. One actin whenever touches the another actin from the opposite side, no more further. This is the maximum contraction. Okay, so they will contract like this. The Z lines are coming closure, so that's why the actin filaments are coming closure, and after that it will carry out contraction. So just see what is happening. This is the M line. M line will be like this only. Okay. Now just see what happened, that we are going to see this myosin is completely overlapped by actins, or actin filaments are coming too much closer that it touches each other from the opposite direction, right? So what we can say that here this zone, what is happening whenever we have contraction, the Z line come closure. Okay, that actually brings the actins together from both the sides towards the M line, right? Which zone will become maximum? O zone become maximum, right? And which zone will disappear? See, this is the H zone, isn't that, where we have only the myosin which is not overlapped by the actin, right? So that's why what will happen? Is it now whenever we have contraction, is it the space present that we can have only the myosin? No, this is all overlapped by the actin, right? So that's why what happened? This O zone become maximum and H zone disappears. Okay, so this is the contraction process. So that's all about the muscle contraction, and how this muscle contraction will occur. This is one very big chapter. We have another chapter, that is the movement and locomotion. In the further chapters we will proceed, but before that you need to know this part clearly. Okay, and whenever we will discuss movement and locomotion, I'm not going to repeat all these things once again, right? So this is all about the contraction process. Hope you have understood. Read too much, read from the book, then see this video over and over again so that you have no confusions left. So this way we will complete the skeletal muscle. In the next video we will see the smooth endocardiac muscle, right? Thank you.